Good day, my dear students. Welcome to the class on flow cytometry. Technology, is it helping us or replacing us as pathologists? Only time will say. This is a university question by itself, and in the world of technical rats, we shall be unraveling some of the newer mysteries. The flow cytometer is a measure of the multiple properties of a cell. It is called as cytometry, the measurement. And as the cells flow in a single column, it is called as flow cytometry. These cells interrupt a beam of laser light and the shadows are picked up by the computer. Microscopy, whereas, is a study of intact cells. This is one of the original flow cytometers designed by Kaminsky way back in 1970, and it was under biophysics. Now you find that whatever subject that we avoided, chemistry, physics and all, are all becoming part of medicine. It is much more difficult to measure non-performance than performance. A beautiful saying by Jenny. Again, a wonderful reference which I would like you people to kindly go to. Though the leukemia has been taken as a prototype in flow cytometry, it will be adding much more to our knowledge. Think of the subheadings. The subheadings will take you to the answer. There are usual subheadings such as definitions, principle, etc. But in this particular topic, I shall emphasize upon the instrument, its general uses in pathology and elsewhere. Also in hematology. What are the problems with a flow cytometer and the description of its usage? A schematic diagram is mandatory for every answer. In fact, it will definitely give the facelift that your answer needs. Coming to the definition, it is a standard laser-based technology for detecting and measuring the physical and the chemical characteristics of a cell or a particle. It is used for rapid analysis, both quantitatively as well as qualitatively. Quantitatively means maybe the number of cells of a particular lineage and the number of particles, etc. Qualitatively is further subclassifying them so that we get a more precise diagnosis. There can be an assessment of the particle size, the granularity, and the complexity within the cells. Fluorescence adds to the armamentarium. The basic principle for flow cytometry shall be optical to electronic coupling system. So always my teacher in physics used to say, there will be one form of energy which is getting converted into another form. For example, heat can be converted to light. So here, optical to electronic. It detects the cells based on the laser scattered by the cells. Now, flow cytometer by itself need not necessarily concentrate only on the cells, but there are other properties also. The chromosomes, the molecules, etc. Always there is a nice history to any of these things. Coulter's law of impedance. It is the principle upon which the flow cytometer is based. This was by Wallace Coulter. And all of you will be knowing something about the Coulter cell counter. It was this that is being applied, as we shall be seeing in the next diagram. And Fulweller was the inventor of this, Mac Fulweller. There are two principles of it. One is the absorption method, another is fluorescence method. It absorbs the light that is thrown on it, and secondly, it can be scattered also. These are the two basic principles. And cytometry or cytometer is the instrument. And what we are getting as a graph can be a cytofluorograph. So this is the probe that we use for any of the instrument, be it biochemistry or pathology. And you find that there is a column of cells that is being taken, later on analyzed and reports given. Two people, only one picture I would be able to get. Louis Kaminsky for the optical scanner. And the next person, Heisenberg, who has won the award for advanced technology. He designed the cell sorter with fluorescence screening. Look at the simplicity with which these people appear and the immense contribution they had made to mankind. The flow cytometer is an instrument that illuminates the cells as they flow. It detects and correlates the signals from the illumination. Beautiful. A couple of things that we people will have to memorize. It illuminates and then it detects and correlates the signals. It has got certain unique abilities. Rapid analysis of thousands of cells, 100 to thousands of cells per second. Also, there are two major principles, measurement of the physical properties and measurement of the antigenic properties, as we shall be seeing. And when it comes to this fluorochromes, chromes are nothing but colors, and fluorochromes are radiolabeled substances, which conjugate with the antigen that might be on the cell surface or within the cell. As a result of which you have got a conjugated chromogenic substance that fluoresces. So it is called a fluorochrome. Always there are a set of colors that have been used and standardized. This I would like you people to kindly memorize. There are more than 60 such substances, but I have projected only 5. Fluorescent isothiocyanate, so FTC is here, rather greenish in color. And phycoerythrin, that's over here, more of yellowish. Texas red, so there is a Texas red over here. Hello, phycocyanin, that is here, more of orange. And pyridinin chlorophyll, that is over here. So, and there is a laser blue also. I would like you people to kindly memorize this so that you will be able to mention the different chromes that are used in a flow cytometer. So this is the German word for it, quite difficult to spell. And it was with this that there was a first patency that was obtained. And it has been developed further by the American Engineering Conference in Automated Cytology. Not necessarily for medicine alone, but for other disciplines. In 1968, there came a flow cytometer, and 1976, there can be a flow cytometry applicable to both. So the word flow cytometry is applicable to both the measurement of it as well as the fluorescence. So this has become more or less an order of the day. It is not just the size of the cell, but also the immunofluorescence. Look at this for an example. So this is a study of a blood sample for the malarial parasite. So it locates that there are some cells which are having the property of showing the malarial parasite as such. And it is ruled out. It has been picked up by means of a side scatter, and scatter means always this particular square over here is considered as neutral or nil. And when it goes beyond either on the y-axis or the x-axis, it is considered to be positive. So here it is positive both on the x-axis and on the y-axis. And this is again picked up, studied with the specific fluorescent antibodies, and it is found to be positive. And autofluorescence has been ruled out. So the message here is it can be used for the diagnosis of malaria at least for the time being. This is the basic principle, the concept, which I shall be repeating any number of times. So there are some cells, and these cells have got surface antigens. There will be specific antibodies against these antigens with which they are mixed. So you find that the antibody gets bound to the antigen. The antibody in turn, it has got a coloring substance or a fluorescent substance. Otherwise, you will not be able to see an antigen-antibody complex. So what happens is, there are the groups of cells which have been treated with these substances. And when they flow through a particular column, the laser beam, they are interrupted by the laser beam, and then the various properties such as the complexity, size, etc. is taken into account, and you get the values. So this is a very simplistic diagram. You people can have in mind whichever suits you, you can draw. So there is a liquid stream. As you are seeing over here, there will be a liquid stream through which it is going. There will be an outer sheath and an inner sheath of this liquid. The cells will be passing through the inner sheath, and that in turn passes through the light beam. What happens over here? There is a light source, there is a lens, and these are the cells which are interfering, and they are being filtered by a series of filters, finally passed on to a detector, and then they produce the images which are thrown on the screen, either for you to record or for taking a print. There is a detector, as you people see, analog to digital conversion. There are two types of scattering. I shall be telling the people this. There is a forward scatter and a side scatter. Dyes, which are used for fluorescence, I have already mentioned. And there is also something called as an amplification system, which again I shall be mentioning. Finally, there is a computer. So you people should be able to recollect all these components. A laser, then a column, an outer sheath, an inner sheath, series of filters, then forward scatter and side scatter, the detectors, the fluorescence that is used for coloring it, the amplification, and finally the computer message. Basically, the flow cytometer is used for three purposes. One is research. Wherever we go, this is now haunting us. Though we wanted to do medicine, to practice, and to treat the patient, research is now haunting us. Clinical practice, of course, and trials. It can be clinical trials or other experiments. For all of these, you find that the basic usage is found in flow cytometry. Even otherwise, this you people shall and will remember. It can be used for cell counting, sorting out the cells, finding out the characteristics and functions of specific types of cells, detecting microorganisms in microbiology. It can be a biomarker detection, protein engineering. This is something beyond us, going towards genetics and so on. Leukemias, measuring the genomes. This again is something new. We shall be visiting it later. So look at the wonderful spectrum of usage. This particular diagram will be very important, and this is the source. I would like you people to kindly go to it. But even otherwise, stay put with me for the next few seconds. So this is a diagram of the flow cytometer and flow cytometry. So there is an exciting laser. There is a laser that is going. And this is a column of cells. You find that there is only a single column that is passing through. As I mentioned, there will be an outer sheath and an inner sheath of fluid, the cells pass through a inner sheath alone. Therefore, only one cell at a time, which interferes with the laser beam. It can either allow the laser to pass straight, which is called as forward scatter, or sometimes it can get reflected sideways, which is called the side scatter. And also you find that there can be a series of filters, filter 1, 2, 3, etc., which will help with the amplification. Amplification is nothing but magnifying it several times, just like your microscope. And finally, as I told you, one form of energy is converted to another, and there is a digital conversion, and this is being finally fed to the computer. This will be the basic thing. So there is a fluid system, there is an optical system, there is an electronic system. This particular diagram alone, if you people understand, it is more than enough for today's class. So, hats off to this one. Again, yet another reference. I would like you people to kindly go to it. So this particular author has given the uses of it in hematology. By flow cytometry, you find that there are different blast cells which are positive for different markers as such. CD34, 45, etc. So you are able to identify a particular group of one and you are able to pick up what kind of a leukemia it is. For example, myeloperoxidase in the case of acute myeloid leukemia. And there can be 17, 117, 13, and 61 for monocytic markers. So myelomonocytic when both are going to be positive. And cell sorters. I would like you people to explain this particular picture. This is what we are doing day in and day out at the cell counter. Look at this picture. There are a group of cells which are large. There is a large nucleus and there is also some kind of indentation. I will not say that they are all equally dark. This appears to be having a much darker nucleus than these. In some, I am able to see pale nucleoli. All these will contribute to the physical properties. These are taken into account and they are assessed, measured, quantified, and qualified. So these are the things: the size, the density, nucleus, the shape, granules, if there are any, the organelles. Now, what happens in this? In a case of fluorescence, there will be different kinds of colors that are being used specific to that machine. So in this, it is a blood sample, of course. I am finding that there are groups of cells and these groups of cells have been given a different color as such. So you find that the blue one is supposed to denote the lymphocyte, the red, the myeloid, the blast cells, whereas the myeloid cells are green in color and the monocytes otherwise. This is a normal sample and the blasts here are just 1.5%. Whereas in a case of a leukemia in this picture, I find that there is a dense cluster of these blast cells and the count goes on to 28%. Obviously, a diagnosis of AML is made. Sometimes you will have a difficulty in making a diagnosis and the flow cytometer comes to our rescue. Not only in hematology, there are other disciplines also where it can be used. So this is the chloroplast structure of a phytoplankton, which is an organism. And look at the appearance of it. This is being studied and this is again the same physics of a single column interfering with the laser beam and there is a scattering of the light, the properties being captured. Now, the signal processing. This is something very strange. In One context I had seen that this is a cell, it has got 7 dimensions. 7 dimensions. Looks like a new movie. But then you people should remember that there will be anterior posterior, there will be the lateral, there can be one oblique and another oblique, and finally there can be a circumferential one. So I have got 7 dimensions that are there. All these are being assessed and captured, and it passes through the series of tests finally onto the computer, through which I can get either a histogram or it can be a dot plot by which I get the values. A very simple one which you people shall and will not only remember but explain. A beautiful diagram as such. But please remember 7 dimensions. And what are the other applications? This is just an introduction. So in the immunophenotyping of lymphomas and leukemias, malignancies, the DNA ploidy and the cell cycle, the CD4, CD8 counting, particularly in HIV, and histocompatibility in a case of transfusion or transplantation, reticulocyte count, paroxysmal nocturnal hemoglobinuria, and also in autoimmune diseases, HLA-B27 detection. So look at the wide spectrum of uses of the flow cytometry. This is only a glimpse of it. The next class, I'll be giving you a complete detail of it. So this again is, in a case of hematology, you can add, apart from hematological malignancies, there can be minimal residual disease, myelodysplastic syndrome, the DNA RNA cycle, etc. And these are also there, and the quantification of the antigens, CD20, 22, 33, etc. A nice table to memorize. Leukemia. I'm not going into the details of the leukemia, but just a word of warning. In a flow cytometer also, it need not always be perfect. The count can be lower than the manual value, or it can be higher than the manual value. Why? Because there can be a dilution with the peripheral blood in a flow cytometer. Therefore, obviously, the count will be low. Sometimes you find that the blast, we find some of the blasts themselves are supposed to be poorly differentiated. Some of the blasts we can very easily say it is a myeloblast. But there will be some cells which are not very clear, and then we call them as poorly differentiated. So such blasts can lack the expression of the surface markers. And also the CD45, this is supposed to be the vital study. And the expression of this also can vary from blast to blast or from cell to cell. Therefore, some cells in that group also can be missed. So this is an important one. And the second one is you find that there can be the NRBCs which can be interfering. Then there is a defect in the separation. Fecal hypec is a solution that is being used for separation. And sometimes the blast cells can be poorly preserved. So what will happen is there can be a increased amount of count in these. Flow cytometers, the general uses we have already seen. More than 1,000 cells or particles per second. And they are separated because of the optical properties. Basically, it is similar to the microscope, only then we are not getting an image, we are getting the values and the shadow of it. And a single cell suspension is needed. Always, when I say that there is a software, the computer is a software, the design is a software, there will be a hardware which is retaining it. For example, in this, there is something that is supporting the sheath of cells. This is one. There is a laser, and then there are the series of optics and the electronics. All this will be the hardware of the flow cytometer. So there is a hardware, there is a software. So what all do you find in a case of flow cytometry? It can be the cell surface marker. There can be intracytoplasmic substances which can be identified. It can be part of the DNA, etc. The cellular processes such as apoptosis, etc. can be captured. Also, intracytoplasmic cytokines and fluorescent proteins within the cell also can be. I would like you people to kindly by heart this. So what are all the various usage of it? You people can look at the diagram and recollect. Even otherwise, by heart this particular one. This gives a near total or a complete list of usage of the flow cytometer. The various things. These we have all seen. Apoptosis is one. Sperm analysis, platelet function, signaling pathways, chromosome, intracellular cytokines, that is over here, gene reporter, etc. So you find that there is a plethora of uses, and I would very much appreciate that you people write not 50 or 60% of it, almost 100% of this particular list. This is what you people are doing with your Sysmex or a Coulter counter. So you find that there will be a forward scatter based on the size of the cell and a side scatter based on the granularity of the cells. So obviously you find neutrophil is one cell that has got the maximum amount of granules. Therefore you have a high value over here. Whereas the least will be the lymphocytes, hardly any granule, and monocytes minimal. And this is again diagrammatically depicted. So there is a forward scatter detecting the size, etc., and the side scatter detecting the granules. So beautiful, simple diagrams I have given you people. Kindly draw them, recollect them, reproduce them. So there can be the other applications in cell kinetics, etc., genetics, molecular biology, animal husbandry also, in veterinary, microbiology, parasitology, and my earlier topic of bioterrorism. I was surprised to find that flow cytometry has got an usage here also. And this is the ancient flow cytometer that was originally designed by Macfield. And his own words are these. And this again is yet another reference. So look at the number of references I'm giving you people. Nothing is my own. Kindly go back to these. And of course, this is a modern flow cytometer. Maybe you people would have seen better versions of these. And there's a probe which is making a suction of it. There is a single column that is taken. The cells are being made to flow in a single column subject to the laser light, and there will be the series of detectors. That can be the light scatters. Finally, there is an image that is over there which is captured onto the computer, which can be in the form of a graph or it can be dot plot. So one or more lasers I had shown you earlier, multiple photodetectors. The signals are amplified. So this is extremely important. More than us just seeing, you find it is amplified and sent to the computer, recorded or printed or displayed as graphs. So this is one of the last diagrams. I would like you people to kindly explain. There is a light source over here. And there is a chamber. Look at the funnel-like thing. It is a chamber. The cells are passing through it. There will be an outer sheath and an inner sheath. And it is the inner sheath through which the single column of cells, the cells in suspension. And they might be having fluorescence over here on the surface. And you find that if there are granules, there is a side scattering, otherwise it passes straight. So this is an indication. See how one simple diagram can denote the entire concept of it. There is a cell, there is an antigen on the surface, there is an antibody, and there is a fluoride. People that spend time looking for faults in others should spend time correcting their own. Maybe it is applicable somewhere. Use of fluidics is over here. So for passing the cells through the laser beam, moving cells one by one, cell sorting, etc., the hardware and the hydrodynamics for the precise positioning of the cells. And the sample is input the speed at which it traverses, and the central sheath should not be mixing with the side. All these come under the hardware, and these are the components which I had explained earlier. My dear colleague, don't worry about what I am doing. Worry about why you are worried about what I am doing. Again, maybe it's applicable somewhere. So this is a summary of it. I would like you people to kindly go through it. There are earlier versions, there is a technical advance, there are modern instruments, there is increase in the number of lasers, the detectors, etc. And they can target particular population of cells, take digital images. And the data is obtained from the samples and mediated by means of the computer. The software is meant for imaging and for the adjustment of the voltage, etc. All the parameters are fed correctly. So this is one beautiful one, again a recollection. My dear student, how is your performance? If you can't measure it, you can't improve it. So I would like you people very much to measure your performance right now. See you in the next class of the applications of flow cytometry. Thank you.